Children, we are going to learn about the life process today. Living organisms have well organized structures. They have organs. Organs have tissues. Tissues have cells. Cells have smaller components, cell organelles, and so on. If the order breaks down, the organism cannot stay alive. Living organisms must keep on repairing and maintaining their structure. Their molecules keep breaking down and new ones forming all the time. The process which performs the maintenance and repair work of cells are life processes. Modes of nutrition means methods of obtaining food by an organism. There are mainly two modes of nutrition, autotrophic and heterotrophic. Autotrophic nutrition is that mode of nutrition in which an organism makes or synthesizes its own food from the simple inorganic materials like carbon dioxide and water present in the surroundings. The green plants have an autotrophic mode of nutrition, while heterotrophic nutrition is that mode of nutrition in which an organism cannot make its own food from simple inorganic materials like carbon dioxide and water and depends on other organisms for its food. All the animals have a heterotrophic mode of nutrition. A heterotrophic organism or heterotroph can obtain its food from other organisms in three ways. So, the heterotrophic mode of nutrition is of three types: saprotrophic nutrition, parasitic nutrition, and holozoic nutrition. Saprotrophic nutrition is that nutrition in which an organism obtains its food from decaying organic matter or dead plants, dead animals, and rotten bread, etc. Parasitic nutrition. The parasitic nutrition is that nutrition in which an organism derives its food from the body of another living organism without killing it. Holozoic nutrition is that nutrition in which an organism takes the complex organic food material into its body by the process of ingestion. The ingested food is digested and then absorbed into the body cells of the organism. It has been found by experiments that the presence of sunlight, chlorophyll, carbon dioxide, and water is necessary for the process of photosynthesis. So, we can say that the conditions necessary for the photosynthesis to take place are sunlight, chlorophyll, carbon dioxide, and water. The photosynthesis takes place in the following three steps. Number 1, absorption of sunlight energy by chlorophyll. Second, conversion of light energy into chemical energy and splitting of water into hydrogen and oxygen by light energy. Third, reduction of carbon dioxide by hydrogen to form carbohydrate like glucose by utilizing the chemical energy. Experiment to show that the sunlight is necessary for photosynthesis. Take a potted discharge plant. Take a thin strip of aluminum foil and wrap it in the center of one leaf on both the sides while leaf is still attached to the plant. Use clips for fixing it tightly. Keep this potted plant in the bright sunlight for 3 to 4 days. Immerse this leaf in the boiling water for few minutes. This will break down the cell membrane of leaf cells and make the leaf more permeable to iodine solution so that it may reach the starch present inside the leaf cells. Plug the leaf in a beaker containing alcohol. Place the beaker containing alcohol and leaf in the water bath. The boiling alcohol will decolorize the leaf. Now wash the colorless or the pale leaf with hot water to soften it and remove any chlorophyll which may be sticking to it. Place the colorless leaf in the petri dish. Drop iodine solution over decolorized leaf with the help of the dropper. Observe the change in color of leaf. The part covered with the aluminum foil or black paper does not turn blue-black, while the rest turn blue-black. The covered part was not exposed to the sunlight, while the rest part was exposed to the sunlight. Therefore, we conclude that sunlight is necessary for photosynthesis. Experiment to show that chlorophyll is necessary for photosynthesis. We take a potted plant like croton, whose leaves are partially green and partially white. The green part of the leaf has chlorophyll, but the white part of the leaf does not have chlorophyll. Place this plant in completely dark place for about 3 days to destarch its leaves. Take out the potted plant from the dark place and keep it in the bright sunshine for 3 to 4 days. Pluck the variegated leaf from plant, boil it in the water for few minutes and then remove its green color, that is chlorophyll, by boiling it in alcohol. The green part of the leaf get decolorized. So we get decolorized leaf. Wash the decolorized leaf with hot water to soften it and remove any chlorophyll which may be sticking to it. Pour iodine solution over the colorless leaf and observe the change in the color of the leaf. We find that the outer part of the leaf that was originally white does not turn blue-black on addition of the iodine solution, showing that no starch is present in this outer part of the leaf. From this observation, we conclude that the photosynthesis to make starch does not take place without chlorophyll. We take a potted plant having long and narrow leaf and place it in completely dark place for about 3-4 days to destarch its leaves. Take a glass bottle having a wide mouth and put some potassium hydroxide solution in it. Take a rubber cork which fits tightly into the mouth of the glass bottle and cut it into the 2 halves. Put a destarched leaf of the potted plant in between the 2 halves of the cut cork and then fit the cork in the mouth of the glass bottle. The upper half of the leaf should remain outside the glass bottle and only the lower half of the leaf should be inside the glass bottle. The potted plant is kept in the sunlight for 3 to 4 days. During this period, the upper half of the leaf gets carbon dioxide from the air, but the lower half of the leaf does not get any carbon dioxide. This is because of the carbon dioxide of the air present in the glass bottle has been absorbed by potassium hydroxide solution and no fresh air can come into the closed glass bottle. Plug the leaf from the plant and take it out from the glass bottle. Remove the green color chlorophyll from the leaf by boiling it into the alcohol. In this way, we get decolorized leaf. Wash the decolorized leaf with the water to remove any chlorophyll which may stick to it. Pour out iodine solution over the colorless leaf and observe the change in color of the leaf. We will find that the lower half part of the leaf does not turns blue-black on adding iodine solution, showing that no starch is present in this lower half of the leaf. From this observation, we conclude that the photosynthesis to make the starch in the leaf does not take place without carbon dioxide. Now, the question arises, how do plants obtain carbon dioxide? The leaves contain large number of small pores called stomata, and the plants take carbon dioxide from air and it enters the leaves through stomata present on the leaf surface. Stomata pore is surrounded by a pair of guard cells. The functions of the guard cells: gaseous exchange and transpiration. The shape of the guard cell in the monocot is a dumbbell shaped, while in the dicot it is bean shaped. Now coming to the opening of the stomata, when the water flows into the guard cells, these cells become curved and causes a pore to open. While when the guard cell loses the water, they shrink, become straight and close the stomatal pore. Now, nutrition in animals. Nutrition in animals take place by the following 5 steps. Ingestion. Process of intaking of food inside the body is known as ingestion. Digestion. Breaking of large insoluble complex molecules into small water soluble simple molecule is known as digestion. Absorption. Process in which digested food passes through the intestinal wall into bloodstream is known as absorption. Assimilation. The process in which absorbed food is taken in by the body cells and used for energy, growth and repair is known as assimilation. Egestion. Process in which digested food is removed from the body is known as egestion. Amoeba feeds on unicellular plants or the animals. Pseudopodia engulfs the prey by forming food cup. The food passes on the food vacuoles, which provide secretion juices to help in digestion. Digested food is absorbed and undigested food comes out from the surface of the body. It is holozoic nutrition. Digestion is a catabolic process. Alimentary canal is a long tube running from mouth to anus of a human being or other animals in which digestion and absorption of food take place. It is also known as gut. The alimentary canal consists of several organs. Number 1. Mouth. Mouth is the uppermost opening of human digestive system, which gives passage for ingestion of food. Second, buccal cavity. Buccal cavity contains teeth, tongue, and salivary gland. Teeth are of 4 types: incisors, canines, molars, and premolars. The function of teeth is chewing and grinding of food. Function of tongue is to taste the food, mixing the food with saliva, and to speak. Salivary gland secretes saliva. Saliva contains salivary amylase that breaks down this starch into the simple sugar that is called maltose. Now the food will enter into the esophagus. The esophagus is a long and tubular structure which serves to carry the food from the pharynx to the stomach. It exhibits peristaltic movement, that is, contraction and expansion. With the help of the esophagus, now the food will enter into the J-shaped muscular bag known as the stomach. The stomach contains gastric juices, that is, pepsin, HCl, and mucus. The work of pepsin is to break down the protein into the simple peptones, while HCl makes the medium acidic so that pepsin can work on it and kills the bacteria that enters into the body with the food. The work of mucus is to protect the inner lining of the stomach. After stomach, the food will enter into the small intestine. The small intestine receives the juices from the liver and the pancreas. The liver secretes the bile juice. The bile juice helps to emulsify the fat as well as makes the medium alkaline. Pancreas secretes the pancreatic juice that contains trypsin, amylase, and lipase. Trypsin helps in breaking down the protein into the peptones, while amylase breaks down the carbohydrate into the simple sugar, that is a glucose, while lipase works on the emulsified fat and turn it into the fatty acid and the glycerol. Now, the food will enter into the large intestine. This large intestine absorbs the excess of water. It also contains the mucus that helps the binding of the fecal matter. The food will now move towards the rectum. Rectum temporarily collects the waste. Then it moves towards the anus. Anus helps in the egestion with the help of the anal sphincter muscles.